All right, so finally, let's look at the second type of termination. And sorry, that should be a number two, not a number one. And this is row dependent termination. So in this case, the row protein is going to be floating around inside the nucleus, and so it's one of those substrates that a specific gene may or may not require. If a gene does require it, then that row protein is going to bind to the RNA, to a certain RNA region, and is going to move upwards towards the 3' end of that messenger RNA strand. As it moves upwards, it's going to encounter a termination sequence that is the same sequence that we see in row independent termination. And that's going to cause the row protein to pause, and then, or that's going to cause RNA polymerase to pause, allowing the row protein to catch up, as we see here in the second section. And then the row protein is then going to unwind the DNA RNA hybrid and bring transcription to an end. So we still get formation of the hairpin loop, but the row protein here is actually the one disassociating the new messenger RNA strand from the DNA strand. So two major systems in termination: row independent does not need the row protein, and then row dependent does need the row protein. All right, so let's talk very briefly about eukaryotic transcription differences. So first of all, the promoter region in eukaryotic systems is going to have a lot more proteins involved in the initiation complex. So we talked about sigma being the main subunit that contributes to the holoenzyme. In eukaryotic systems, we're not only going to have the general or core promoter region, we're also going to have regulatory promoters that can either speed up or slow down how fast transcription takes place. And in eukaryotic systems, there's actually multiple polymerases, like we talked about earlier, that have very different functions depending on what type of RNA is being produced. In initiation, differences that we see with eukaryotic cells is that transcription is going to be initiated when the basal transcription apparatus that consists of RNA polymerase plus transcription factors assembles on the core promoter and becomes an open complex. And then finally, in termination, one of the major differences that we see in eukaryotic cells is that for different eukaryotic RNA polymerases, they're going to use different methods of termination, even more so and more complex than row independent and row dependent. So those are three major differences associated with eukaryotic systems compared to just our prokaryotic systems. So as we look at our eukaryotic promoters, we tend to have, if this is our start site right here, we've got core promoters that are going to be upstream, and then we have regulatory promoters that are going to be downstream, as well as upstream. So that Tata box is still very much in place, but in eukaryotic systems, we usually don't see it until minus 25 above the start site. And then we see another consensus sequence at minus 35. And then, so that's going to be our core promoter region. Our regulatory promoter region is going to be even further upstream than the core promoter. And this is going to allow further regulation of transcription, because that's one pathway that we don't ever want to get out of control, because if it happens and it can't be shut down, that's where we start to see tumors being formed and accumulation of tissue that's really not supposed to be there. All right, so in eukaryotic systems, the promoter region is going to be carried out by accessory proteins, and they are going to recruit very specific RNA polymerases. So these transcription factors become very important in this process. So different transcription factors will come in, bind to the holoenzyme, and we'll also see activator proteins come together and help bind to sequences in the core promoter region. And this is all going to contribute to um, the effectiveness of a messenger RNA production in the transcription process. So just realize that the promoter region is going to be enhanced with the use of transcription factors and transcriptional activator proteins. The core promoter is going to then work to alter the rate of transcription, so how fast it actually happens. So it can increase it or it can decrease the speed of it depending on the protein that it interacts with. And then enhancers are really important in eukaryotic systems because they will work to usually increase the rate of transcription, and they can be either upstream or downstream of the transcription start site. So again, we've got core promoters and regulatory promoters controlling when transcription actually takes place. The efficiency of transcription can then be enhanced by mediator or transcription factor proteins. We can get activator proteins that are going to help the transcription process, and then we can also get enhancers that will bind to regulatory promoter regions as well.